Hello, my name is Marom Bikson, and I'm going to be talking about what are subthreshold mechanisms in neuromodulation and brain stimulation, including spinal cord stimulation and really all applications of neuromodulation. Subthreshold mechanisms is something maybe that a lot of people have sort of heard about, but are not entirely sure what they mean in the context of making neuromodulation effective and making brain stimulation effective. So I'm hoping that this talk will clarify that, and I've kind of divided up into four sort of simple parts to digest. And you can get the slides here. So what is subthreshold neuromodulation? And I think it's good to introduce that concept in terms of what is supra-threshold neuromodulation. Now, typically we don't call most neuromodulation interventions like DBS or SES or TMS as supra-threshold. We just say they are what they are. And so we're presenting here supra-threshold in contrast to this other thing I'm going to be talking about called subthreshold neuromodulation. But most neuromodulation, most brain stimulation is supra-threshold. And what that means is that the intervention, first of all, is cooked from pulses, right? This would be a series of pulses at some prescribed frequency, like 10 hertz or 100 hertz with some pulse width, pulse shape. And that is what is being delivered to the targeted brain region. That could be a brain region in the cortex or the spinal cord or in the periphery, right? And then the idea is that this train of pulses is then leading to a train of action potentials of neurons in the targeted region. So simplistically, you would say if I'm putting in 100 hertz pulses, I'm going to be producing 100 hertz rate of action potentials in a group of target neurons. So it's all about pacing. It's all about driving action potentials, and so it's clear why pulses really matter because of this correspondence between a supra-threshold pulse above threshold and then the generation of an action potential. Now, the downstream consequences of neuromodulation, if it's treatment of depression, treatment of movement disorders, rehabilitation, whatever else happens is secondary to the pacing of these action potentials, right? So the benefits derive from that. And therefore, when we are applying neuromodulation interventions, when we are designing neuromodulation interventions, we're always thinking about how are we cooking up these pulses, right? How are we deciding on things like pulse frequency, pulse width, in order to drive specific types of action potentials, in order to produce specific kinds of outcomes. And so conversely, when we're innovating, you want to invent a new supra-threshold neuromodulation technique that has to be around a new pulse recipe. Now with subthreshold neuromodulation, we can use pulses, but we can actually use a wider set of waveforms. We can do things like sinusoidal, noise, or even DC, right? Just turning it on and leaving it on. And that energy is still applied to a targeted brain region with neurons in it, but now what we're thinking of is not so much the pacing of these cells as their polarization. And then this polarization leads to a change in how that brain region works, how it processes information, and that in turn underpins the clinical outcomes. So you can see there's a bit of a difference here, first of all, starting with the kind of recipes that we're using to create waveforms, but also there's a core difference where with subthreshold stimulation, we're thinking about polarization and changing processing. And this whole talk will be explaining what we mean by that. And as it turns out, therefore, in subthreshold neuromodulation, when we're thinking about how a technique works or we're trying to innovate something new, we again have to start with the waveform, but now we have a larger constellation of possible waveforms and then a different mechanistic pathway. And what I'll try to sort of convince you in the background is thinking about subthreshold neuromodulation instead of the more conventional supra-threshold neuromodulation. It opens up new vistas for how you might understand neuromodulation and also how you might design neuromodulation interventions. Now, a lot of the data I'll show you initially is using a subthreshold waveform of a very long pulse, so a pulse that is turned on and could be left on even for as long as a second. So it's on for so long, it could be described as sort of like a DC, right? Because it's on for such a long period of time. And the reason I'm going to base some of the introduction on long pulses, DC-like pulses, is I think it helps a lot with intuition of how all subthreshold neuromodulation will work. So it's like a conceptual building block. And so here we are applying a slow long pulse, right, a DC stimulation to a population of neurons, and they have in this case shown in a cartoon, like this characteristic architecture where you have morphology, where you have the somas represented in triangles, and there's a little bit of a basal dendrite up top, and then an apical dendrite down below. And in this particular example, we're putting the positive electrode, the anode, at the top, and the negative electrode at the bottom, so that the electric field current is moving in the downward direction, washing over these neurons. Now, what will it do as it washes over these neurons? Well, it will polarize them. So what you're looking at here is experimental data from a rat brain slice using optical imaging with voltage-sensitive dyes, and all the details are going to be in the, you can dig into them in the papers that are cited down below. But what you can see by eye is that when the direct current, this long pulse, is turned on, the cell polarizes. Its membrane potential changes, and it stays polarized for as long as the pulse is there. But if we look at it a little bit deeper, you'll notice that the polarization is not the same across different compartments of the cells. The soma, for example, in this particular stimulation modality and direction, is being hyperpolarized. So is the top basal dendrite, but the lower apical dendrite is being depolarized. Its potential is moving in the opposite direction. So right away we see that how much polarization you see and which direction it's going to be on depends a lot on the compartment. We can't say that this stimulation is a depolarizing stimulation. We can't say that it is a hyperpolarizing stimulation. We can only say that some compartments are hyperpolarized and some compartments are depolarized, and even the amount of polarization you see is going to vary across compartments. So that's one key idea. Now, you'll also notice that the polarization sort of looks like the stimulation, meaning we turned on the stimulation and the compartments polarized, and they stayed polarized for as long as we left on the stimulation, and then they went back. And as it turns out, if you apply other waveforms like a sinusoid, the polarization will look like a sinusoid. So when we talk about subthreshold stimulation, it is generally the case that the polarization of the cells looks like the waveform of the stimulation that you're putting in. But you'll notice that there is a bit of a lag. So when we first turn on that stimulation, there's like a charge up of the cell membrane, and then when we turn off the stimulation, there's a discharge. And in this case, the time constant of that charge up and discharge is about 8 milliseconds. And this is another important idea. Finally, there's no action potentials. The polarization that you're seeing here, which is shown in a percentage as far as voltage sensitive dyes, but it corresponds to about a millivolt of polarization, is not enough to generate an action potential. So we're producing a real measurable polarization, but we are not driving action potentials because this is a subthreshold field. And so this introduces the first concept, which is the notion of coupling constant. And it should be polarization instead of politicization. But the polarization goes something like this. It's a very simple equation. You need to first start with the notion that you have a cell, but we have cells with different compartments. And for each compartment, we're going to assign a sensitivity to stimulation or a coupling constant, right? So this would be something like 1 millivolt of polarization, right, per volt per meter of applied field, right? It is how sensitive, how much polarization you get in that compartment per the amount of field applied. Now the field applied is the second term, right? And by the way, this is going to be compartment specific, right? Different compartments on the same cell are going to have a different a coupling constant, right? And then we have a field that is applied, right, of a certain intensity. We control that. We can apply a lot of stimulation. We can apply a little bit of stimulation and so on. And then the polarization that we get is simply the electric field strength in volts per meter times the coupling constant, right, which is in millivolts per volt per meter, right? And sometimes the coupling constant will be referred to also as the polarization length, because of the resulting unit. So let's go through an example. Let's say we have a compartment of some cell in the body, and its polarization length, its coupling constant, is 0.5 millivolts of polarization per volt per meter of applied electric field. We apply an electric field. We're stimulating the body. Let's just say it's 10 volts per meter. So now the cell is exposed to 10 volts per meter. Well, very simply, we're going to predict that that neural compartment, the specific one we're talking about here, not all compartments, the one we are focusing on in that one cell will polarize by 5 millivolts. And if you double the amount of intensity, 20 volt per meter, you will get 10 millivolt of polarization. If the coupling constant was double, you can see how it's a very simple linear relationship. And so this coupling constant is the first conceptual concept of subthreshold neuromodulation. Now, the question is, well, how big are these coupling constants? Because it seems like it's going to matter a lot. If they're near zero, we're not going to see much polarization, right? If they're very large, we're going to see much more polarization. So the question is not just how big they are, but also how big could they get, right? So if we're looking across the brain, there's different compartments on different cells. How sensitive can the brain be? And this question has to be divided, first of all, into considering somas and dendrites, because as I've already hinted, they have different coupling constants because of the morphology of the cells. And we also have to consider a third compartment class, which is axons. So we ask the question, how big is the coupling constant for somas, for dendrites, and for axons? And this is something that I and other people have worked on for, I would say, well over a decade. And in 30 seconds, right, what did we learn? Well, we learned that the coupling constant of somas is about up to, I would say, 0.2 millivolt per volt per meter. And this is for the biggest, most asymmetric kind of cells. So that's how much we predict the somas of neurons in the brain will polarize to an electric field. It's harder to get, and this was done with intracellular recording of the somas. It's harder to record from axons, but there are ways to do that, including through the recording of blebs. And using this type of analysis, we concluded that terminals will polarize up to 0.8 millivolt per volt per meter. So more, right? The terminals are more sensitive than the soma. The question of dendrites, it turns out, is still harder to address experimentally, and I'm actually putting a reference here to a modeling paper, and I'll talk more about that, but they're somewhere around 0.5 millivolt per volt per meter. So basically you're seeing it's less than 1 millivolt per volt per meter. And again, these are the most sensitive cells with the most sensitive compartments. So we're looking at the sort of the upper limit. And so that's summarized right here. For somas, just remember about 0.2, dendrites about 0.5 millivolt of polarization per volt per meter, and axon terminals seem to be the most sensitive, polarizing up to 0.8 millivolt per volt per meter. Now, these coupling constants are not that big, right? What I mean by that is, in order, if we think that a cell needs to be depolarized 10 millivolts to fire, right? 10 millivolts to bring it up to action potential threshold, you can imagine the size of electric fields that you'll need in order to produce that. You need electric fields of about 100 volt per meter in order to make cells fire. Conversely, if we apply a lower intensity electric field, let's say we just apply an electric field that is just 10 volt per meter, right? The soma is only going to polarize 2 millivolt, and the axon terminal will only polarize 8 millivolt, and this is not necessarily enough to produce action potentials. And this brings us back to the notion of subthreshold neuromodulation. Subthreshold neuromodulation posits that you don't need so much fields that you generate action potentials. The polarization is enough, all right? And the coupling constant tells us how much polarization you're going to get. But what does that polarization do? And that's the second thing, right? So the second part of this conceptual part is that notion that that polarization produced by electric fields that is not enough to generate action potentials can still change how cells work, right? Polarizing somas and dendrites and axons is not nothing as far as how the brain works. It will change how the brain works. And specifically, our work is focused on the notion that it will change synaptic processing, right? Synaptic processing is the bread and butter of how brain works, of how all brain works, how pathology manifests. And so the hypothesis that we've sort of centered a lot of our research on is that when you apply an electric field and you polarize somas a little bit and dendrites a little bit, and terminals a little bit, you will change synaptic efficacy, right? How connective connected different brain regions are, the strength of the synapse from one cell onto another cell. And an important corollary of this idea is that we're not generating sort of de novo action potentials that then sort of slam through these synapses. The brain is already active. The action potentials are already coming. The synapses are already active. What we're changing is how effective those synapses are. So this is an essential idea of subthreshold modulation of synaptic activity. We are not generating de novo synaptic activity, we're modulating ongoing synaptic activity, right? This is the second concept. So how does the polarization that we talked about in part 1 of these different compartments lead to changes in synaptic processing? And here we go again. This is going to be 15 years of research in just a minute. And again, I'm putting a lot of references below that can, for people who really want to dig into this a lot more. The way most of these experiments were designed is, first of all, there is ongoing synaptic activity, right? The brain, the animal model that we're using is already active, and you can measure synaptic activity using different things like intracellular recording of an excitatory postsynaptic potential, or an extracellular recording of a field excitatory postsynaptic potential. So that's what you're seeing here, right? And the only thing you need to understand is that the size of that you have an ongoing synaptic activity. That's what each one of these blips represents, and the size of that blip represents the strength of the synapse, how strong the coupling is from the pre to the postsynaptic size, and you can see that it's roughly stable after the first few pulses. Now we apply on top of that, right, with the ongoing activity, a long DC pulse, which of course produces a polarization. And we measure the change in synaptic efficacy, and you can see here by eye, right? Then when we apply a long DC pulse for the period indicated by red, the synaptic efficacy is increased, right? The ongoing activity, the ongoing coupling is suddenly boosted. There's a stronger connectivity. And then when we turn it off, it goes back to baseline. Now, there's many variations on this theme, but this is a very robustly reproduced idea. Sub-threshold fields that produce a little bit of polarization, right, will change synaptic efficacy. Now, that being said, there is a lot of subtlety to how this is done, and the reason why it's subtle is because you are actually polarizing different compartments, and depending on which compartments you polarize and which pathways you are considering, you could see different effects. Generally speaking, when we're polarizing axon terminals, when we hyperpolarize the terminal, we increase coupling. Now, this may seem counterintuitive, but in fact, when you hyperpolarize the terminal, you increase the drive for calcium influx, you actually enhance neurotransmitter release. So this is actually a known idea. So fields that hyperpolarize terminals boost synaptic efficacy. With dendrites, it's a bit more subtle, but often hyperpolarization also favors synaptic efficacy, and this is because you are actually increasing the driving force for currents as they come in and as they arrive at the soma. And again, I'll refer you to the papers for more detail. It is not always this case. There are situations where dendrite depolarization favors synaptic efficacy. With somas, the story tends to be rather simple. The somas is sort of where the decision of whether or not to generate an action potential or not is generated. It's where all that synaptic information is being coalesced and processed. And there generally, soma depolarization increases synaptic efficacy. And if you want to put one number on all of this, and of course it will vary from system to system, roughly, you'll see a 1% change in synaptic efficacy per volt per meter of electric field applied. So that a 10 volt per meter electric field applied to a brain region will produce a 10% change in synaptic efficacy. Okay, well, going back here for a second, I just said soma depolarization favors lowering action potential threshold. Now, up to now, I wasn't talking about action potentials, right? I said subthreshold stimulation doesn't produce action potentials, right? But then this brings us to the next idea that if you modulate membrane potential, if you modulate synaptic efficacy, you will change the likelihood of generating action potentials, right? And this is this third concept, part 3 of sort of subthreshold concepts, amplification. Ultimately, if the neuromodulation is going to be affecting brain function, if it's going to be treating depression or epilepsy or pain, it does at some point need to change how action potentials are being generated. That is the currency of the brain. But what we're doing with subthreshold neuromodulation is we are not generating the action potentials, right? We're not driving in these pulses. What we're instead doing is we're changing how action potentials are generated in the context of ongoing brain activity. And there's several great resources on this. This is one, and let me walk you through an example that sort of makes the idea clear. Imagine the cell starts initially at minus 70 millivolts, and then this is without electric fields or electrical stimulation. The cell's just sitting there at minus 70, and some synaptic input shows up. And I've illustrated that here simplistically as a straight line, and that synaptic input brings the cell up from minus 70 to minus 50, and minus 50 just happens to be the action potential threshold, and then you get an action potential, and then the cell recovers. Okay, so this is without electrical stimulation. Now, let's assume that the synaptic input was not quite as strong, right? It was only enough to polarize the cell by 19 millivolts, right? So it brings the cell up to minus 51 millivolt, just shy of action potential. So a little bit makes a big difference, right? No action potential in this case. Now let's add electrical stimulation. Let's apply a low intensity DC pulse, just enough to produce a 1 millivolt worth of depolarization. That means the cell goes from a resting membrane potential of minus 70 to a resting membrane potential of minus 69. Now we apply the same 19 millivolt excitatory synaptic input. It is now enough to produce an action potential. So now we can see that in the context of ongoing brain activity, in the context of ongoing synaptic input coming in, right, a small difference, right, in the membrane potential of the cell, right, bringing it up a little bit, can make the world of difference in whether or not an action potential is generated or not. And it turns out that, in fact, in the brain, your cells are not sitting at minus 70. They tend to be all up and around action potential threshold, and they're constantly deciding whether or not to generate action potential. So it's important to understand when we think about brain function, we're thinking about cells constantly almost or moving through action potential threshold, and it's in this sort of primed system that we are now adding low intensity polarization, and therefore we're expected to change how these action potentials might be generated. So again, a small polarization, in this case, let's say a millivolt, can produce a big change. Okay, now let's go back to the situation where without electric fields, the input was enough to produce a action potential, right? Now let's apply a field. Well, let's think about what would happen. Well, the membrane potential is stepped up. This is by 1 millivolt. The same input comes in, and you can see that in this case, well, we're still going to get an action potential, but it's going to come earlier. So even in situations where the ongoing activity is sufficient to generate an action potential, subthreshold fields, low amounts of polarization, still make a difference by influencing timing. And it turns out that timing is a very important signal inside the brain. And what we showed in this publication, in this sort of simplified situation, is there's a very simple equation that will tell you how much change in timing you will get. So the amount of timing change you will get in, let's say, milliseconds is equal to the amount of polarization produced by the field divided by the slope of the ramp, which means the more narrow the ramp, the bigger amplification you see as far as change in timing, right? So that's why as the title says, spike timing amplifies the effects of electric field. And this is just one mechanism that has been identified of how the nonlinear action potential based processing of neurons in the brain, right, leads to an amplification of subthreshold fields. The brain is already nonlinear, the brain is already very active, and therefore it is not surprising that you could find situations where a small polarization makes a big difference. And again, I want to remind you that this polarization on itself was not enough to generate action potentials, right? When we first applied it, 1 millivolt, it's not supra-threshold, but it is not inert, and that's the point. Now, when we talk about ongoing brain activity, part 4, the final idea I want you to be aware of is brain oscillation. So brain oscillations, our brains are always oscillating. All parts of the spinal cord is oscillating. And there's been a lot of work done on the significance of these oscillations. All I want you to take away here is when you have oscillations, well, the cells are active, right? And here I've sort of illustrated in triangles excitatory cells, and you have inhibitory cells as well, in sort of lighter color, and they're sort of coupled with these synapses, and they're all active, right? But the thing about oscillations is they're not just randomly active, they're active in a particular pattern, right? That's why you see the oscillations, for example, interneurons, excitatory neurons, interneurons, sort of a pattern back and forth. And so you have not just activity, but a network of cells tightly coupled together, generating an emergent phenomenon, which is the oscillations. And now on top of this highly active, highly coordinated activity, we apply an electric field. And what we and others have shown is that these network oscillating systems are very sensitive to small perturbations, right? Because they have this feature, first of all, that all the cells are near threshold, right? Because that's what the oscillation is. It's bringing cells in and out of threshold. But on top of that, if you affect one cell, right, you're going to affect its neighbor as well. And so you have the second level of amplification that comes from the fact that the network itself is so tightly coupled. And again, I'll refer you to this paper and others that have looked at it. This is one experimental result. This again is data from rat brain slice, where you have an ongoing oscillation. Gamma in a rat slice is around 35 hertz. You can see the power in the spectrogram. The long DC pulse is turned on from 1.5 seconds to 3.5 seconds and then turned off. And you can see, first of all, when it's first turned on, there's this massive increase in power. You can see that in the time series below as well. And then as long as that field is on, we have a higher level, an increased level of gamma oscillation power. When we turn off a field, you get this sort of rebound inhibition, and then it comes back. Now, there's a lot to be said about these results and similar ones. I just want to point out one thing, which is that the field on its own, the stimulation, does not generate oscillations, or it doesn't generate gamma oscillations, right? The brain is generating a type of oscillation, and the field is modulating that ongoing oscillation, right? That is a common theme in subthreshold stimulation. You are modulating ongoing activity, and it turns out that when you have network oscillations, they're very, very sensitive to electric fields. But again, the response you get can only be understood once you consider what is the ongoing activity, right? If you have ongoing alpha and you apply field, you'll modulate that. You have ongoing gamma, you will modulate that. And what we've shown in this paper and others have shown as well is despite the apparent complexity and nuance of this, you can actually model this very reliably using rather simplistic models that will explain exactly how subthreshold stimulation and why networks are so sensitive to subthreshold stimulation based on their coupling type features. Okay, those were the 4 concepts. Now I want to bring this sort of back to a particular intervention, which would be the neuromodulation of pain, and a particular mechanism, which is the gate control theory of pain. This is a theory that has been around since the 60s, developed by Melzack and Wall, and I'm just going to say very briefly that the high-level notion is that you have painful input coming from the periphery, passing through the spinal cord, and going through these, what Melzack and Wall called T cells, before finally being transmitted to the brain. So pain needs to be processed through the spinal cord. Now you also have additional input coming from the periphery. These would be what they described as large diameter axons. These would be carrying non-painful information. Now, this is also transmitted to the brain, but it makes a stopover in the spinal cord. And the key to Melzack and Wall's idea is that when these non-painful fibers make a stopover in the spinal cord, right, they inhibit the transmission of the painful signals, which is where you get this sort of notion of, if you rub it, it feels better, right? If there's painful input coming from a particular dermatome and it's being processed through the spinal cord, right? We now rub it. we activate the large diameter fibers, right? And then that has the effect of inhibiting the transmission of the painful signal through the same brain region. So that's a very high level idea. And what I want you to take away from this here is that it's synaptic based, because all brain processing is synaptic based. And so both the excitation coming in from the large diameter axons, right, that's an excitatory influence. So is an excitatory influence from the painful axons. And then the gate, the closing, is an inhibitory synaptic signal. Now, since Melzack and Wall, others have developed sort of more complicated, the neuroscience of in general has expanded. The neuroscience of pain has become more sophisticated. Nonetheless, I think at a very high level, this remains the case, that you have inputs and you have gates, and it's all synapses, right? All right. So spinal cord stimulation was based on Melzack and Wall's idea. And with spinal cord stimulation, the notion is that you are stimulating the white matter axons, right, as they're heading up the central transmission line, right? But that central transmission line will antidromically go back into the spinal cord in order to close the gate. Now, the gate exists in the gray matter, right? So we're stimulating the white matter, right? That's the traditional notion with spinal cord stimulation. We're stimulating the white matter in order to activate the gray matter. And the pain relief, right, according to gate control, does happen in the gray matter, right? That's where the magic happens, right? So we're stimulating the white matter, which is producing incidentally a tingling, a paresthesia, right? Because that's that central transmission. But it then goes back the other way where it closes the gate, right? So traditionally, spinal cord stimulation is understood to stimulate the large diameter axons, closing the gate. But might it also stimulate the gray matter directly? Could it also stimulate the very same cells that are responsible for the closing of the gate, right, as a alternative or as additional mechanism? Well, certainly for the past 50 years, that has not been the predominant theory. White matter stimulation, axon stimulation, has been the predominant theory. Why is that? Well, one of the reasons is that it was considered for a long time that for spinal cord stimulation to work, you needed to produce the tingling, the paresthesia, right? And that's consistent with stimulating of these large diameter sensory axons. You need to get them going, which inevitably produces the paresthesia, right, in order for them to then be activated, go back into the dorsal, the gray matter, the dorsal horn, where the gate is closed. Turns out that this is not necessarily the case. We now have subparesthesia therapies. The other reason why people were pushing back against the fact that we were stimulating the gray matter, the actual location of the gate, right, was that when you stimulate, you're stimulating sort of in the epidural space. So you have epidural space, then you have your cerebrospinal fluid, right? Then you have your white matter, and then you have your gray matter. So the gray matter is farther away from the stimulation electrode. And by virtue of being farther away, the intensity of electric field in the gray matter is less than that in the white matter. And so if you increase the intensity to the point that you have enough intensity in the white matter to drive action potentials, you may not have enough intensity in the gray matter to drive action potentials. So if you're focusing on supra-threshold mechanisms, you're going to say axons go first, right? And they're being primed and driven well before we get to intensity levels that will drive action potentials in the gray matter. So that was another reason that people said, well, if the electric fields are higher in the white matter and we need to produce action potentials, well, then it must be white matter stimulation. But of course, subthreshold stimulation changes the calculus, first of all, of whether you need action potentials to be driven directly or not, right? And secondly, how big of a field you need. And so the alternative hypothesis is that spinal cord stimulation generates electric fields inside the gray matter itself, in the dorsal horn of the spinal cord, that are not enough to generate action potentials. They're what we're going to call subthreshold, but they're nonetheless enough to change the processing of the gray matter in such a way that the gate closes. And this is an idea that, in fact, has been around for as long as the gate control theory of pain, and many people have been working on this idea. But in my own case, sort of, having worked on subthreshold mechanisms for several decades, it's very natural to translate that kind of thinking, right, to the gate control theory of pain. Now, related to that is another concept which was recently developed, which had to do with closed loop stimulation and had to do with recording what happens when you stimulate. So people discovered that when you stimulate the axons, right, you make them fire, that activity is then conducted and you can record it. And so this was something called an ECAP, right? And I'm going through this very quickly. I'll refer you to the paper, right? So you stimulate, you produce a white matter response called the ECAP. We discovered in a rat model that there's something else, something that we call an ESAP. Now the ESAP, right, the N1 and the P2, right, or would be and getting into the N2 would be the traditional ECAP, right? But rising from the N2 going into this other thing labeled the S1 is the ESAP. And what we showed in this paper is that the first component, the ECAP, does correctly reflect the transmission of white matter axons along the spinal cord, but that the ESAP is generated from inside the spinal cord, is generated from the gray matter specifically, right? So the ESAP, right, is an evoked synaptic activity potential. It is synaptic in general. It is actually recording the activity, the activation of the gray matter by stimulation, right? So we can record from the epidural space the activity, the synaptic activity being generated inside the gray matter of the spinal cord during stimulation. And there's this concept of reciprocity, which says, broadly speaking, if you can record something like a synapse with an electrode, you can stimulate that synapse with an electrode, right? It's still like going in and out. And so the fact that you can record synaptic activity being generated from inside the gray matter of the spinal cord in an epidural electrode argues indirectly that with that epidural electrode, you can stimulate the same processes generating that synaptic activity. So that's a very high-level way to approach it. Now, to try to put some sort of more details on it, we went back to coupling constants, right? We said, okay, spinal cord stimulation is producing electric fields, not just in the white matter, but in the gray matter. How much polarization is it going to be producing and where? So we relied on computational models. This is a model of spinal cord stimulation. You have the lead placed over the spinal cord. You have an electric field that is higher. You can see that in panel D. It's higher in the white matter. It's lower in the gray matter. But we discovered, well, not that much, right? It's a factor of 2 drop off, not a factor of 100 or a factor of 10. Right? So there's still a substantial electric field in the gray matter. And while 12.5 volt per meter, this is for each 1 millivolt applied, right? So 1 milliamp applied. So for example, 2 milliamp applied would generate 48 volt per meter in the white matter and 25 volt per meter in the gray matter. And 25 volt per meter may not sound like a lot from the perspective of supra-threshold stimulation. From the world of subthreshold stimulation, that's a lot. And inside the spinal cord gray matter, we consider different types of cells or cell types that have been classically associated with processing in the spinal cord. These are inhibitory cells, and classically some of them have been associated with gate control itself. The preprint is listed below, but to make a long story short, we found that the cells that were most sensitive to polarization, to subthreshold polarization, were these very elongated islet type cells, and this largely derived from the fact that they were so big. And we predicted that at their terminals, you would get about 3.5 millivolt of polarization per milliamp of SES applied. So for example, 2 milliamp SES would produce 7 millivolts of polarization, which again, from the perspective of subthreshold, is a big number, all right? We also consider the impact of pulse width. So whereas 3.5 millivolt would be what you'd get with a very, very long pulse, as the pulse gets shorter and shorter, you will produce less polarization. And this goes all the way back to that concept I mentioned at the beginning about the time constant of polarization. Using some simplifications, we were able to figure out that depending on the time constant of the cell, we considered between 1 and 16 millisecond time constants, and depending on the pulse width, you're going to get a different amount of polarization. But generally speaking, the longer the pulse, the more polarization you're going to be seeing. Now, as far as things like pulse density or pulse frequency, how many pulses you have per second, if we think of the polarization as happening when the pulse is on, you can think of increasing pulse density or pulse frequency as increasing also the amount of time that the cell is held in a subthreshold polarization state. Now again, there's interaction between pulse width and so on, but generally, right, as you're packing in more pulses, as you're going to sort of dense pulse patterns, you have the opportunity to sort of add up the amount of time that the cell is spent in a polarized state. And if the cell is held in a polarized state, how it processes synaptic activity will change. Another interesting point is how we think about targeting. So I mentioned the islet cells were the most polarized, but the most polarized of the islet cells were between the electrodes, not under them. While other types of cells, such as these very sort of vertical type cells, would be most polarized under the electrodes as well. So when we start thinking about subthreshold polarization, right, as a mechanism of action of spinal cord stimulation, we might also start thinking differently about which cells are being stimulated where, and therefore where we would put our stimulation electrodes. And so that is just a hint of how you would approach a problem like spinal cord stimulation, which is very traditionally thought about as supra-threshold stimulation, from a subthreshold stimulation, and also a hint, and I know I covered it very briefly, a hint of how if you start thinking about it in terms of subthreshold stimulation, right, not only do you move to a completely different region of influence, right, from the white matter to the gray matter, you might also start thinking differently about how you titrate things, like pulse width and electrode position. So this brings me back full circle, right? In the case of supra-threshold stimulation and in the case of subthreshold stimulation, in both cases we are synthesizing waveforms. Whereas supra-threshold stimulation, it's really about pulse to pulse base. In subthreshold stimulation, we don't need to use pulses. And even when we are using pulses, we can think of a more integrative type of polarization. Supra-threshold stimulation drives action potentials. Subthreshold stimulation modulates ongoing activity, modulates synaptic function, modulates network activity. And that is how subthreshold stimulation can have profound effects on brain function. And to conclude, if you're approaching things from a sort of a subthreshold mindset, or from a sort of a superthreshold mindset, or from a subthreshold mindset, you may actually lead to different conjectures of how stimulation works, how you might optimize stimulation, and therefore I believe subthreshold neuromodulation thinking can lead to new therapies, more effective therapies for everything that neuromodulation is used for. Because as a final point, when you are doing stimulation at supra-threshold intensities, all the subthreshold mechanisms are still there, right? So that means that all neuromodulation activates subthreshold mechanisms as well. So thank you for your attention.